Welcome to the May edition of AJP's Video Table of Contents, or From the Editor's Desk. The May issue brings a wealth of great reading, which includes 4 research articles, several original research papers, and our first cardiovascular physiology guidelines papers. We hope this will be one of a series of papers that will provide a consensus opinion in various areas of cardiovascular physiology. Each guideline paper will be authored by experts in their respective fields and will be accompanied by a prolonged discussion in one of our podcasts. These are major works that, in many cases, are over a year in the making. I urge you to read and listen to these informative new articles. So let's get started and see what's inside this issue. The first guidelines paper by Hart et al. Provides a detailed discussion of best practices in the recording of sympathetic nerve activity in humans and animals. The authors discuss both technical issues and pitfalls to consider when recording in various environments, including in conscious and anesthetized animals. An important aspect of this paper is the discussion of the various ways sympathetic nerve activity can be quantified and different ways of normalizing the data in animals and humans. For those interested in setting up an animal lab or engaging in microneurographic techniques in humans, this article is a must-read. Nabi et al. Review the role of immune function in giant cell arteritis in the aorta and its medium-sized branch vessels. They discuss how CD4 T cells, macrophages, and dendritic cells build granulomatous infiltrates that injure the vessel wall and elicit a maladaptive response to injury. Under physiological conditions, immune checkpoints provide negative signals to curb T cell activation and prevent inflammation-associated tissue destruction. Harrington et al., in a novel review, discussed the use of psoriasis, a chronic inflammatory skin disease associated with a greater risk of early cardiovascular events, as a suitable human model to study the pathophysiology of inflammatory atherogenesis in humans. The authors have been utilizing this human model in a large prospective cohort study and summarized their approach and results of utilizing this human model to study inflammatory atherogenesis. The review simultaneous multimodal imaging of several vascular beds using FDG, PET, and CT, FDG, PET, and MRI, and coronary CT angiography, as well as cardiovascular biomarkers to better understand how modulation of inflammation may impact cardiovascular diseases. In an interesting review by Ungvari and colleagues, the increasing prevalence of multifocal cerebral microhemorrhages, CMHs, also known as cerebral microbleeds, is discussed. CMHs are associated with rupture of small intracerebral vessels and are thought to progressively impair neuronal function, potentially contributing to cognitive decline, geriatric psychiatric syndromes, and gait disorders. Clinical studies show that aging and hypertension significantly increase the prevalence of CMHs. CMHs are also now recognized by NIH as a major factor in Alzheimer's disease pathology. Finally, they examined potential treatments for prevention of CMHs based on the proposed model of aging and hypertension-dependent activation of the ROS-MMP axis and discussed critical questions to be addressed by future studies. Guo and co-workers investigated the effect of angiotensin 1 through 7 on the development of cardiac hypertrophy and identified the intracellular mechanisms underlying the action of Ang1-7. Ang1-7 significantly attenuated Ang2-induced cardiac hypertrophy and perivascular fibrosis in rats and in cell culture. This protective effect of Ang1-7 was significantly attenuated by pretreatment with A779, a mast receptor antagonist, mitotempol, mitochondrial targeting superoxide scavenger, as well as blockade of SIRT3, a deacetylation acting protein, by viral vector-mediated overexpression of SIRT3 shRNA. Moreover, Ang1-7 increased FOXO3A deacetylation and SOD expression, and these effects were blocked by SIRT3 shRNA. Thus, activation of the Ang1-7 SIRT3 signaling pathway could be a novel therapeutic strategy in the management of cardiac hypertrophy and associated complications. Chen et al. sought to identify if microRNAs are dysregulated in response to angiotensin II-induced hypertension in the kidney and whether H2S donor GY4137 could reverse the microRNA alteration and kidney function. Wild-type mice were treated without or with Ang2 and GYY4137 for 4 weeks. Blood pressure, renal blood flow, and resistive index were measured. Ang2 treatment significantly increased blood pressure, decreased blood flow in the renal cortex, and increased resistive index and reduced renal function. These effects were ameliorated in mice treated with GYY4137. MicroRNA 129 was downregulated in Ang2 treated mice and upregulated following GYY4137 treatment. These data suggest that downregulation of miR-129 plays a significant role in Ang2-induced renal inflammation and functional outcomes, and that GYY4137 improves renal function by reversing miR-129 expression. Study by Sharma et al., the role of angiotensinogen in the paraventricular nucleus, or the PVN, of rats with heart failure was investigated to identify a likely microRNA that regulates angiotensinogen. Overexpression of miR-133A in NG108 cells resulted in a 1.4 and 1.5-fold decrease in angiotensinogen and angiotensin type 1 receptor mRNA levels. Luciferase reporter assay performed on NG108 cells confirmed that miR-133 bound to the 3' UTR of angiotensinogen. Further, they observed a 1.9-fold decrease in miR-133 expression with a concomitant increase in angiotensinogen and AT1 receptor expression within the PVN of heart failure rats. Restoration of miR-133 abrogated the enhanced renal sympathetic nerve activity responses to angiotensin microinjections within the PVN of heart failure rats. These results reveal a novel and potentially unique role for miR-133A in the regulation of angiotensin within the PVN of heart failure rats. This may potentially contribute to the commonly observed sympathoexcitation seen in heart failure. Lal et al. Determined if there is a role for decreased VEGF-B in diabetic cardiomyopathy. They found that of the VEGF-B located in cardiomyocytes, there is a substantial and readily releasable pool localized on the cell surface. The immediate response to high glucose and the secretion of endothelial heparinase is the release of this surface-bound VEGF-B, which triggers signaling pathways and gene expression to influence endothelial cell autocrine action and cardiomyocyte paracrine effect survival. Under conditions of hyperglycemia, when VEGF-B production is impaired, a robust increase in VEGF receptor 1 expression ensues as a possible mechanism to enhance or maintain VEGF-B signaling. Through this bidirectional interaction between endothelial cells, which secrete heparinase, and cardiomyocytes, which release VEGF-B, this growth factor could provide the diabetic heart protection against cell death and may be a critical tool to delay or prevent cardiomyopathy. Thank you again for watching from the editor's desk. As I've mentioned before, we're constantly soliciting your feedback about the journal, your likes and your dislikes. We want to publish the most relevant work for the cardiovascular physiological community, and we need your help. I hope you will send us your best work, and we look forward to talking with you again in June. Have a good month.